So students, after completing the fertilization, now we are moving to the next topic, that is post-fertilization events. What are the post-fertilization changes we are just going to see, or we are just going to observe? Normally that process we have observed earlier also when we were discussing about the normal concept of the sexual reproduction, and here once again now it's time to understand the post fertilization changes occur in the plants. So what are the post fertilization changes? The first of all, when the fertilization gets complete, whatever the floral parts are there, these floral parts like that, sepals, petals, stamens, stigma, style, they all are just getting shred off. Whatever the floral parts are there, all the floral parts are getting shred off. Shred off means what? They are witter off. They are just now they are not the part of the flower. In many of the but in some of the flowers, the sepals remain persistent. Sepal remain persistent means to say the sepal is present always with the flower and with the fruit only, even after the fertilization and even embryonic development. The example of the plants where the sepals remain persistent with the embryo, that is tomato, brinjal, chili, etc. So normally what happens, all the floral parts are getting shriveled off, all the floral parts are getting wither off, but in some of the cases the sepal is present along with the embryo. Even after the embryonic development, they are present with the fruit also. So the example of that kind of the flowers, that kind of the plant is tomato, brinjal and chili. One very important thing, that ovary is always persistent, and that ovary is finally just getting converted in form of the fruit. That is what the normal condition or normal conversion is there. Ovary always present and ovary is just going to convert in form of the fruit. So what happens, now we are just going to understand one by one, step by step, what are the basic changes that occurs in the flower after the fertilization. So just see, there are four important events are just going to take place. The number one, development of endosperm. Number two, development of embryo. Number three, development of seeds. And number four, development of fruits. So the four events are there. The first one, the endosperm development. Second one, the development of embryo. And third one is the development of seeds. And fourth one is the development of fruits. So now we are just going to move on to the first one, that is development of endosperm, how the endosperm is going to develop and what is the importance of endosperm. So one by one we are just going to move on. So first step or first change is development of endosperm. So first of all, we must understand what is the endosperm. If you have remember, in the previous lecture during the fertilization, only the plants or angiospermic plants are having what? Double fertilization and the triple fusion. Triple fusion, the polar nuclei are going to fuse with the second male gamete and they are just going to produce a triploid structure. So endosperm is a triploid structure which is having responsibility to store the nutrient or the reserve food material and which will be provided to the embryo for the embryonic development. Are you getting my point? So endosperm is a triploid tissue which stores the nutrient for the further embryonic development. So this is required for the embryonic development. So if endosperm is present therein, so the embryonic development will be better. If it is not there, so that will affect the development of embryo also. So what happens, how the endosperm is produced? If you have remember, yesterday only we have just seen after the triple fusion, the polar nuclei fuse to the second male gamete and they form the triploid structure. That triploid structure is called as primary endosperm nucleus. That primary endosperm nucleus is getting converted in form of endosperm by the cell division. What is there? The primary endosperm nucleus is getting converted in form of endosperm by the cell division, and basically the division is now being involved, that is the mitosis type of division, not the meiosis. So on the basis of development how the endosperm is going to develop, the endosperm may be classified into three basic categories. Now the important portion begins. On the basis of development, the endosperm can be categorized into three basic categories. The number one is called as nuclear endosperm. The nuclear endosperm is the most common in the angiosperm. More than 56% families of angiosperms are having the nuclear endosperm. Along with those, along with that, dicots for polypetally. Capsula, the important example, the capsula. Again and again we are saying that capsula is the example where the embryonic development has been studied. Callitrops, malva, citrus, they all are the example of the nuclear endosperm. What happens there in the nuclear endosperm? The nuclear endosperm is developed by the free nuclear division. Free nuclear division. The endosperm, the nuclear endosperm is developed by the free nuclear division. It is developed by free nuclear division. What is the meaning of free nuclear division? The free nuclear division stands for when the nucleus divides again and again, but without the cell wall formation. No cell wall formation occurs, only the nucleus division is occur. Are you getting my point? So developed by the free nuclear division without the cell wall formation, so that is called as nuclear endosperm. So what is nuclear endosperm? Which is developed by the nuclear division, the consecutive continuous nuclear division, but without the cell wall formation. So it is clear to you all. So means we can say if a nucleus is going to divide again and again and going to produce the endosperm, so that kind of endosperm, the nuclear endosperm, is called as multinucleated endosperm. So this is prenuclear endosperm or multinucleated endosperm. And one more thing we need to take care of that, even after many of the nuclear division, it will have to convert finally in form of the cellular type of endosperm, because at last the cell wall formation will be there, and when cell wall formation occurs, so that will be considered as a cellular endosperm. So this is what. So initially the nuclear divisions are there without the cell wall formation, that is called as the nuclear endosperm, and that will be a kind of multinucleated structure, and the cell wall formation will occur at the last, and finally it will get converted in form of the cellular form. So it is clear to you all. Now just see with the help of that diagram. Here in the diagram I am just going to show you the nucleus is there, and nucleus is going to divide again and again. Many of the nucleus are being produced here without forming any of the cell wall around them. The final cell wall is produced outside. So that is what the cell wall, and inside the nucleus is there. So now this endosperm is considered as the free nuclear endosperm or the multinucleated endosperm. So is it clear now? So moving on to the next type is the cellular endosperm. Cellular endosperm is the property of gamopately, nymphaceae and araceae. Gamopately, nymphaceae and araceae. Cellular endosperm is the property of gamopately, nymphaceae and araceae. What is specific, or what is the difference between the nuclear and cellular endosperm? Just remember, in case of nuclear endosperm, the nucleus divide again and again without the formation of cell wall, and here in case of cellular endosperm, each nuclear division is supplied with or accompanied with the cell wall formation. Are you getting my point? Each nuclear division is accompanied with the cell wall formation. Whatever the nuclear division is there, the nuclear division will be followed by the cell wall formation. Just see here in the diagram only. What happened here? The nucleus is divided. The nucleus is going to divide, and after nuclear division, the cell wall is being formed. The blue color is denoting the cell wall. So here nucleus and cell wall, nucleus and cell wall, nucleus and then cell wall, nucleus, cell wall means what? After each and every nuclear division, what will happen? The cell wall formation occur. So now the third type of the endosperm based on the developmental pattern is called as the halobial endosperm. What is that? This is halobial endosperm. Halobial endosperm is the property of family Halobi. Halobial endosperm is the property of family Halobi. Halobial endosperm is a kind of intermediate stage between the above two. Intermediate stage between the above two means what? What happens? After the first division, the cell wall formation occur, and later on there is no cell wall formation observed. Are you getting my point? The cell wall formation occurs only after the first division, and later on there will be no formation of cell wall. That is the property of Halobi family. That is why this is referred as what? Halobial endosperm. Are you getting my point? Only once the cell wall formed, and then no cell wall formation. So no cell wall formation, again and again nuclear division. So first one is nuclear endosperm, and second the cell wall formation is there. So that means this bears the intermediate stage between the nuclear and cellular endosperm. Just to pay attention on the diagram here. What happened in this case, you can see easily Here the first division occurs. After first division the cell wall formation occurs, and after the cell wall formation the nucleus is continuously dividing again and again, but there is no formation of the cell wall around these nucleus. So this is what an intermediate stage. So all these three types are very much clear to you all. In the nuclear endosperm, what happens? The nuclear endosperm develop by the free nuclear division without formation of cell wall. Cellular endosperm will develop the division will develop with the nuclear division followed by the cell wall formation, and the halobial, that is the intermediate, where what happen, the first time cell wall formation occur, and after that there will be no cell wall formation occurs. So after these three basic type of endosperm, nuclear, cellular and the halobial, now we have some specific properties of the endosperm, or we can say some specific endosperm. The specific endosperm, the first group or first type of endosperm is called as ruminant endosperm. Ruminant endosperm is the property of family Anonaceae, Aristolochiaceae, Myristiaceae, Ericaceae, Rubiaceae. The best example to understand that kind of the endosperm is the custard apple and the betel nut. What is specific in that endosperm? The endosperm is having some of the ridges or furrows on the external surface. Are you getting my point? The endosperm of these families, or endosperm of some of the plants, is having ridges or the furrows on their external surface. You can easily just correlate the example of the custard apple. If you can find out the custard apple in your imagination, you can see the custard apple is externally ridged. Again and again the ridges are there. Those ridges are actually the ruminant endosperm. Now the next one is called as the mosaic endosperm. Mosaic means irregular pattern, an irregular pattern of distribution. So sometime what happen, the two different color of the patches, or two different colored patches are present there in the endosperm. Sometime what happen, two different colored patches are present or appear there in the endosperm. Such kind of the arrangement is called as the mosaic endosperm. Are you getting my point? Mosaic endosperm means what? Two different colored patches are present there in the endosperm. Example for that is maize and tomato. Is it clear now? The next one is the horny endosperm. Horny endosperm means the endosperm, if it is surrounded with a thick wall that is made of the deposition, or that is made because of deposition of the hemicellulose. Are you getting my point? If the endosperm is surrounded by a thick wall due to the deposition of the hemicellulose, then it is called as horny endosperm, and the example for that is family Apiaceae. Now very important term we are just going to discuss, that is xenia. Very important term and important for the NEET examination. The part what we are discussing, that is important for the NEET examination. What is xenia? Xenia term is given by the Falk in 1881, and he has reported the event there in the maize plant. So first time it was reported in the maize plant. What is xenia? Xenia is a kind of event where the pollen grains of the foreign agencies, or the pollen grains, are directly influencing the development of the endosperm. Are you getting my point? Here normally the endosperm development is the internal process, but sometimes what happens, the pollen grain influences the external morphology of the endosperm. How the endosperm will behave, how the endosperm will looks like from the external surface, that is what the external morphology. So what is xenia? Xenia is the event where endosperm's morphology is externally the external morphology of endosperm is being affected by the pollen grain, and the direct involvement of pollen grain is being reported there in the external morphology of pollen grain. That was observed there in the maize plant. If you have seen, if you are just able to imagine, the maize plant is having two types of the endosperm. Some of the endosperm is yellow colored endosperm, and some of the endosperm is the white colored endosperm. Yellow colored endosperm is more common since it is dominant, it is regulated by the dominant gene, and somewhere the white colored is also there, so that is regulated by the recessive gene. So whenever the dominant and recessive genes come together, so that time they are just going to control the color of endosperm. So this is what the genetic event present there in the pollen grain. So yellow colored endosperm in the maize plant is the effect of pollen grain on the external morphology. Now next one is the metagenia. After xenia, the next one is metagenia. What is metagenia? Metagenia is reported by the swingle in the plant Phoenix dactylifera in 1928. Getting my point? It was reported by the swingle in Phoenix dactylifera in 1928. Xenia and metagenia is having somewhat differences there. In xenia, what happened? The pollen grain is affecting directly to the external morphology of the endosperm, but in case of metagenia, pollen grains are going to affect or influence the morphology of external layers of the embryo sac. Means they are just going to affect the appearance of the color of the seed. They are just going to affect the shape of the seed. They are just going to affect the size of the seed, maturity of the seeds, fruits, everything they are just going to affect. Are you getting my point? What is xenia? Xenia is the effect of pollen grain onto the morphology of endosperm, but metagenia is what? Metagenia is effect of the pollen grain on the external tissues of the embryo sac. Means perisperm, fruit walls, seed coat, everything, seed shape, seed size, maturity, everything will be affected by what? The pollen grain. So this is what called as the metagenia. Now some more points I'm just going to make you clear. If I'm just going to provide a note, means what? This is very much crucial points for your NEET examination, your final examination. Listen, some specific cases are there along with the above cases. The coconut plant, very crucial question, very unique question. Repeatedly the question has been asked there in the examinations. The coconut is considered as having two types of endosperm. The coconut is having two types of endosperm. The endosperm is the two types. One endosperm is present on the external surface, that is a solid endosperm, and another one is present there in the inner surface or inner side, that is the liquid endosperm. So you knows very well, the solid one is the kernel of the coconut and the liquid one is the coconut milk that we are using for getting nutrition. Are you getting my point? The coconut has two types of endosperm, the outer one and the inner one. Outer one is the solid endosperm, that is kernel, and the inner one is the liquid endosperm, that is coconut milk or coconut water. What is the specific behind that? The solid endosperm or kernel is a kind of cellular endosperm, while the liquid endosperm is a kind of nuclear endosperm. It is a kind of nuclear endosperm. Nuclear endosperm you knows very well. So, and you knows very well one term we have used earlier also, when plant cell is there, so that time the multinucleated cell can be considered as the syncytial cell. So this is also the coconut milk is also considered as the liquid syncytial. And sometime the question is there in very commonly the question is asked why the coconut milk or coconut water is used as the nutrient. So now you are having the answer thereon, because coconut water or coconut milk is the rich source of endosperm nucleus. So this is what, so what we are using when we are using the kernel, so that time we are using the cellular endosperm, and when we are using the coconut milk, that time we are using the nuclear endosperm. Second important thing, the endosperm is found to be absent somewhere. So in that cases they are directly able to absorb the nutrient from the peripheral tissues. So where we can just found the endosperm is absent? The family Archidaceae, family Podostomaceae and Trapezae. The three families are there, Archidaceae, Podostomaceae and Trapezae, where the endosperm is found to be absent. The diploid endosperm we have already seen during the monosporic development of embryo sac. The specific case is there Oinothera type. If you have remember Oinothera type of the embryo sac. So in that case the endosperm is the diploid endosperm. Otherwise endosperm is triploid endosperm. So here in case of Oinothera, that is diploid. And one striking feature you just remember, in gymnosperms the endosperm is haploid in nature. In gymnosperm the endosperm is haploid in nature. Why this is so? Because the endosperm is formed without before the fertilization. Why this is so? In gymnosperms the endosperm is haploid in nature. Why this is so? Because this is being produced before the fertilization. It is being formed before the fertilization. So I think now this is very much clear to you all. And remember, all these points are very much crucial when some of the time the questions may be there to calculate the number of chromosomes. That practice I will just give you after the completion of the topic or at the end of the topic. So till now what we have learned: the specific type ruminant endosperm, when ridges or furrows are present on the external surface. Mosaic endosperm, two different colored patches are appeared there in the endosperm. Horny endosperm, the hemicellulose thick wall is deposited there. Xenia, the effect of pollen grains on the embryos, on the morphology of endosperm. Metaxenia, when the external layer of the embryo sac are being affected by the pollen grain. Then coconut milk is having the liquid endosperm, and the kernel is having the solid endosperm, that is cellular, and the liquid one is having the nuclear one. Endosperm is absent in some families. Diploid endosperm present in the Oinothera, and gymnosperms are always having the haploid endosperm because it is produced before the fertilization. So after having the endosperm or the study of endosperm, now we are very much sure or very much clear about that the endosperms are having the source of reserve food or energy there in the plants. What source of energy or reserve food there in the plant. So how they are just getting the energy from the other parts of the embryo sac? So they are just driving the energy from the other part of embryo sac, and basically the part of embryo sac which is getting the part of embryo sac which is providing energy is the parenchymatous cell, which is present over there. So to drive the nutrition from the other part of the embryo sac, like the parenchymatous tissues or the cells present in the chalazial region and the nucellus, the endosperm itself develops some of the structures which drives or absorbs the nutrition from the peripheral tissues of the embryo sac. So these structures are the outgrowths projected outward from the endosperms to absorb the nutrient or the food or the reserve food material from the other tissues of the embryo sac are called as endosperm haustoria. So endosperm haustoria means the outgrowth of the endosperm which is used to absorb the nutrition thereon. So how many types of the endosperm haustoria is there? On the basis of the reasons from where they are just driving the nutrition or from where they are just originating. So first type of the haustoria is called as the chalazial haustoria. As per the name, it is very much clear to you all. The chalazial haustoria means the outgrowth developed from the chalazial end and which drives the nutrition. Micropylar haustoria. The micropylar haustoria is the haustoria originated from the micropylar end. Micropylar chalazial means what? When it is produced from both the end. So where we can repeat the chalazial haustoria? In the family Nymphaceae and the Araceae. Where we can report the presence of the micropylar haustoria? In family in Impatiens only. It is found there in the cellular type of endosperm, and micropylar chalazial type of haustoria is present there in family Boraginaceae. Now students, we have completed the concept of the endosperm. Here endosperm, you just have observed that endosperm is a triploid tissue which is used to store the energy and provide the energy for the embryonic development, and having three different types: cellular, nuclear, and the halobial. What are the basic points you have just seen? Now I am just going to provide some more thing about the concept of that. What the important thing is that the questions are there. Sometimes the questions may be asked related to the ploidy levels of the endosperm cells or till the cells whatever we have produced till now. So whenever the questions are there, so you just take care of that. Whatever the seeds will be produced, the seeds will be always diploid in the nature. The endosperm is always triploid in the nature. The zygote is always diploid in the nature. Megaspore is always haploid in the nature. Microspore is always haploid in the nature. So once again I am repeating, the ploidy of the seed is diploid. The ploidy of endosperm is triploid. The zygote is diploid. The megaspore is haploid. The microspore is haploid. So here I have just written one sentence. The questions related to the chromosome number. You just take care of that. Sometime the questions in the question paper is there. Just find out to calculate the number of chromosome present in endosperm, to calculate the number of chromosome present in the seeds are found, just calculate the number of chromosome present in the embryo or the zygote, whatever the question is there. They will provide some of the raw data. Suppose they are just going to ask the question, the number of chromosome present in the embryo of a plant is 26. So what will be the number of chromosome in its endosperm and in its gamete, like that. So that time you have to just go through that. Go through that means what? I'm just going to provide some examples there on.
Start.
Suppose the question is there, the number of chromosomes in endosperm of a plant, endosperm of angiosperm plant is 36. So they may ask to calculate the number of chromosome in seeds, in gametes, or in the zygote, or may ask to confuse you in embryo also, or they may provide the number of chromosome in any of these structure, and they will ask to calculate all other to calculate the number in all other structures. So like that. So here if it is 36, so what will be the answer? You just find out 36 there in endosperm. So endosperm is triploid in nature, so what will be there? N is equal to 36 divided by 3. So what is the haploid number of chromosome? That is 12. Now you are having easy answer. In case of seeds, what will happen? Seed is diploid in nature, so seed is 2n means what will be there? 24. If gamete is there, so gamete is n means what? The number of chromosome will be 12. Zygote is diploid in nature, so zygote will have 2n means 12 into 2, so that is 24. Megaspore haploid in nature, gamete haploid in nature, so that will be n only. The microspore n only. And one more thing, sometime they are just going to confuse you. They are just going to provide the concept of the meiocyte. They are just going to give the number of chromosome in a meiocyte of a plant. Meiocyte of a plant, like in a meiocyte of a plant 24 chromosome. I'm just going to provide the same answer. Meiocyte of the plant is having 24 chromosomes. So what will be the number of chromosome in all these structure? So meiocyte is diploid cell, you knows very well. So that has to undergo in the process of meiosis, and by meiosis it will form the gamete. So gametes are haploid. So what will be the number of chromosome in the gametes? Usually 12. Now you can calculate whether it is in endosperm, is in megaspore, is in zygote, is in seeds, or is in embryo. So is it clear to you all? One more situation that is more complicated there. Sometime they are just going to ask to calculate the number of chromosome. Just listen, to calculate the number of chromosome instead of angiosperm, they are just going to ask to calculate the number of chromosome in an endosperm of gymnosperm plant. Now the situation gets changed, because in case of gymnosperm, the endosperm is, you knows very well. Now we have discussed endosperm is haploid in nature. So what will happen? Every scenario is getting changed. Why the endosperm is haploid in nature? We have seen the endosperm is being produced before the fertilization. That is why this is haploid in nature. So you have to take care. The question is either for the angiosperm plant or is it for the gymnosperm plant. If you are not able to differentiate between these two, so you will always suffer from the question. So your work is that to get the practice of such kind of the question, just change the number of chromosome. You just take the example of zygote and find all the number of chromosome. You just take the example of the gamete and find the number of chromosome in all the structure, in gymnosperm also, in angiosperm also. But this is my request, whenever you are just solving the question, you have to go with the reading again and again. You have to find out the key points of the question, whether the question is pertaining to the angiosperm or question is pertaining to the gymnosperm. And this portion is very crucial, very important in CBSE as well as the NEET examination. So just take care. If you are just going to complete this one, means you are able to solve a question of 2 marks there in the CBSE examination. So just take care of that. How you have to solve, you can understand. And even this question is important. Somewhere the question may be asked like the ploidy level of these structures. Are you getting my point? So now we have completed the endosperm development and we have just completed the first phase of post fertilization change. The first phase of post fertilization change is the endosperm development. The second phase is the embryonic development that we will discuss in the next lecture. Till the time, thank you.